uh, Dr. Gonzalez, Bio 310, Nazareth College. I wanted to get to question number 15 in the homework to try to show how to solve that. We're not going to have time on Monday to do it, so I'm making this video. Okay, so let me go ahead and share screen and walk. We're going to walk through this. All right. So, okay, so this is problem 15, and this is, I think, the hardest problem in the homework. And this is asking us, this is, okay, so this question, they're providing us with a genomic sequence of two parents and then of their child. And it's asking us to try to identify any new mutations that have arisen um, uh, in this sequence that are not found in either the parents. So a new mutation that occurred during replication, probably as part of meiosis. Okay, so first, let's understand what we're looking at here, what this figure is representing. Okay, so we have mother, father, and child. And now the way that it's kind of set up, it looks like a double-stranded DNA molecule. So, but it's not. And you can tell that it's not because the sequences are very, very similar. Okay, and we know that double-stranded DNA molecules have to be anti-parallel and complementary to one another. So that's not what we're looking at. What we're actually looking at is two pieces of DNA from two separate chromosomes. Okay, so let's call this chromosome 5. Okay, so let's say that this copy M1 from the mother is a strand of DNA that came from chromosome 5 in her genome, and the copy M2 came from the homologous chromosome 5 in her genome. Okay, so we're looking at a homologous piece of DNA on two separate chromosomes. So we're looking at alleles of a specific DNA sequence. Okay, so if this is a gene, then we're looking at two different alleles of this gene. Okay, and we know they're different because if you look close, you can see there's one SNP right there between those two sequences. Same thing with the father. So let's say it's chromosome 5. So one copy from one chromosome 5. And now we're looking at the same sequence, the homologous sequence from the other chromosome 5, the homologous chromosome 5. And now these went into the child after meiosis and fertilization. And so the child has two copies of chromosome 5, and those are the two relevant sequences that are homologous to the other ones from the parents. All right, so how are we going to solve this? Okay, so I'm going to show you the way that I solve it, and then if you have a different way or a faster way, let me know. I would be pretty cool, but I think the way that I did it works, and I'm not sure that you can get much faster. So let's take a look here. My solution involves two steps. Okay, first, compare the two sequences in the child to each of the copies in the parents and determine if one of the copies is an exact match to one of the parents. Okay, because then if that's true, then we know which parent that copy came from, and we know that it does not have any new mutations, and then we're only concerned at that point about the other copy, and that makes life a little bit easier. And then once we find one that is not an exact match, then we find the parent's copy that it came from that has the least number of changes. And so we're applying the principle of parsimony here that we learned about. Uh, I don't know if we do it in 103, but we definitely do it in 105. We do parsimony. And so parsimony, the principle of parsimony is that the solution with the least number of changes is the most likely solution, because as each change, it requires some probability to change. And as you increase the number of changes, you decrease the probability of all of those changes happening. So yes, that is the product rule. So parsimony, we're going for the highest probability, meaning that we need the least number of changes. Okay, so first step, let's take both of these sequences and compare them to both of the ones in either parent. So easy way to do that is I visually I broke it up and aligned them physically so that they could be compared directly. So the first thing is zoom in real close and look at one strand at a time because it gets confusing if you try to look at both strands at the same time. Okay, look at one strand at a time and see how that compares to both strands of the mother. Okay, so let's look at this first strand, CAG, CAG, and I'm comparing the first strand here to the first strand here. And I'm looking to see if either one of these is perfect match. That's my first goal. So I got CAG, CAG, ATT, GCT, looks good so far. Oh, and we have a problem here. This is a C. So there's a SNP here relative to the first strand here. So that's not a perfect match. Now let's look at that first strand to the second strand, and there's a SNP, there's another SNP here relative to that strand. So this strand here is not a perfect match to either one. Okay, so we're going to keep moving and let's look at the other strand. Remember, we're trying to find a perfect match so that we can exclude that from our trying to solve this problem. Okay, so CAG, and I'm doing second strand to first strand, and then we're going to do second strand to second strand. So I'm second strand to first strand. CAG, CT, okay, second strand to first strand. There's a SNP there, so it's not a perfect match to the first strand. So I'm going to go on to the second strand, and it is looking good. Okay, TT, GCT, TDD, okay. Oh, we have a problem here. This G is not found in that strand. So this strand is not a perfect match to that strand either. So what we know now is that neither of the child's strands is a perfect match to either of the mother's strands, all right? They both have some SNPs relative to the mother strands. So we can do that same thing for the fathers. Again, we're looking for a perfect match. So first strand to first strand, and then first strand to second strand. So let's walk down this CAG, C up. We have a SNP right there. There's an A here where there's a T in the first strand. Okay, so that's not a perfect match. Let's look at the second one. And it's the same situation. Actually, we have a SNP even earlier, CAG here, whereas this is the CAA. So this first strand is not a perfect match to either of the fathers. Now, the last strand, we're going to compare that to the first and the second. Okay, let's compare it to the first, CAG, CTGA, TT. Okay, looks good, looks good. Oh, it is perfect. So this strand right here is a perfect match to the father's F1. It's a perfect match to the father's F1. So now we can say that there is a high likelihood that this strand came from the father and is donated from that chromosome 5. So let's say this strand is father F1. Now, where did that strand come from then? Did the child get both strands from the father? No, right? So if strand 2 here is a perfect match to father's F1, the first strand of the child must therefore have come from the mother. So now we're reducing our the problem to a single strand comparison between strand 1 of the child to both strands of the mother. Now we can start looking for which one of those has the least number of differences. Okay, so strand 1, let's go back to the mother and we'll do a comparison. Okay, so let's zoom in here. So I've got SNPs identified in red, but we'll look at that in a second. So I've labeled this on the side of the father. Let's look at that. So I've labeled this as from mother, and the second strand is F1. So that's from father, perfect match. Okay, so from mother, so let's go over and look at mother, and let's do another pairwise comparison is what it's called. So we know second strand is F1, so we're going to ignore that one and just look at first strand and compare first strand to both of the strands of the mother, okay? Both of the strands of the mother and see which comparison has the least number of differences, okay? So let's look at the top strand. Okay, we got CAGCA, so that's good. GATTGC, okay, perfect, until we get there. That's the one difference. And then TGAG. Okay, so if we compare it to M1, there's one change, all right? Now let's compare it to M2. CAGCA. So there's one change, right? And then we keep going and it's all good there. And there's a second change. Okay, so if we compare it to M2, there are two changes. If we compare it to M1, there is one change. So by applying the principle of parsimony, we can say, or we can infer, that this first strand came from the mother's M1 with one new point mutation here at this position, change of an A to a C. Okay, so I labeled this M1 with a SNP, and this is F1, perfect match to F1. So yes, so labeled here, and just back to the zoomed up kind of larger picture. The first strand is, the second strand is a perfect match to F2, the fathers, and the first strand is a match to M1 with one change. So more likely that it came from M1 with a single change than it came from M2 with two changes. Okay. All right, so that is how to solve that. I lost my zero there. Okay, so I will post this video onto YouTube, and if you have any questions, please let me know. We will continue with chapter 2 on Monday. Okay, have a good weekend.